Hierbij open ik deze academische zitting waarin Ellis Nelissen het academisch proefschrift getiteld Soluble guanylate, cyclase as a novel target for cognition enhancement in het openbaar zal verdedigen. Mag ik u verzoeken een samenvatting te geven van uw proefschrift?
Geachte prorector, leden van de corona, familie, vrienden en collega's. In de komende 15 minuten ga ik jullie iets meer vertellen over mijn proefschrift getiteld Soluble guanylate cyclase as a novel target for cognition enhancement. Of in het Nederlands het eiwit guanylatieklase als een nieuw target voor het verbeteren van het geheugen. En daarmee wil ik beginnen met de hersenen en hersencellen. Hier zien jullie een kaart van het wegennetwerk van Nederland. Met een aantal hoofdwegen en heel veel vertakkingen. Dit is een complex netwerk waardoor wij makkelijk met de auto van A naar B kunnen reizen. In de hersenen hebben wij ook een complex netwerk. Alleen, dat is nog veel complexer. Maar wanneer we dan inzoomen, dan zien we in één keer iets wat veel meer lijkt op de wegenkaart van Nederland. In de hersenen moeten wij namelijk ook dingen van A naar B verplaatsen. Alleen in dit geval is dat informatie. Zo kunnen we bijvoorbeeld herinneringen opslaan of herinneringen terughalen. En dus is het belangrijk voor ons geheugen. Alleen wordt dit niet opgebouwd uit wegen, maar wordt dit opgebouwd uit individuele hersencellen, neuronen genaamd. En deze neuronen die moeten onderling communiceren om zo die informatie door te geven en dat netwerk te vormen. En dat communiceren gebeurt op de plek van de synaps. En deze synaps is dus heel erg belangrijk voor onder andere het geheugen. Maar hoe werkt deze synaps nou precies? De synaps werkt op basis van een neurotransmitter. Jullie kennen misschien bijvoorbeeld dopamine of serotonine als bekende neurotransmitters. Nou, in dit geval is het eerder glutamaat voor het geheugen. Dus wanneer er dan een geheugenstimulus of een stukje informatie in hersencel 1 komt, dan wordt er een neurotransmitter losgelaten in de ruimte tussen de twee hersencellen, de synaps genaamd. En deze neurotransmitter die kan vervolgens binden aan receptoren op de tweede hersencel. Deze zijn er speciaal om die neurotransmitter op te vangen, waardoor dat er in die tweede hersencel allerlei processen gaan lopen. Zo kan er stikstofmonoxide worden aangemaakt, dat dan vervolgens SGC, Soluble guanylate cyclase, stimuleert. SGC produceert dan cyclisch GMP en dat gaat dan vervolgens de cel vertellen wat het nou met de zojuist verkregen informatie moet gaan doen. Er kunnen receptoren, neurotransmitterreceptoren naar het oppervlak worden verplaatst. Er kunnen eiwitten worden aangemaakt. Maar het stikstofoxide is een gas, dus kan ook vrij in de omgeving rondbewegen, waardoor het naar die andere hersencel terug kan bewegen. En daar via SGC de loslating van, of de afgifte van neurotransmitter kan versterken. Zo kan dus zo'n geheugenstimulus via SGC specifiek zorgen voor meer neurotransmitterafgifte, voor meer receptoren op het celoppervlak van de cel, of voor de aanmaak van eiwitaanmaak, zoals bijvoorbeeld meer receptoren, maar ook weer groeifactoren, waardoor een simpele hersencel zoals deze een complexe hersencel zoals deze wordt. En daardoor is er een betere signaaloverdracht en daardoor hebben wij een goed geheugen. En hier staat dus SGC centraal aan. Maar SGC is een complex eiwit. Het heeft namelijk een ijzergroep gebonden. En deze ijzergroep is noodzakelijk, want dit is waar stikstofoxide bindt. En doordat stikstofoxide aan die ijzergroep bindt, kan SGC cyclisch GMP produceren. Maar in een ziekteomgeving kan het ervoor zorgen dat die ijzergroep verloren gaat en dan hebben we een ijzervrij SGC. Hier kan stikstofoxide niet aan binden en hierdoor kan het dus ook zijn, wordt dit eiwit inactief. Tegelijk kan die ziekteomgeving er ook voor zorgen dat er minder stikstofoxide aanwezig is, waardoor het ijzergebonden eiwit ook geen cyclisch GMP meer kan produceren. Dit zijn twee verschillende mechanismen met hetzelfde resultaat. Minder cyclisch GMP-productie en dus een slechter geheugen. Dit wil ik dan ook op twee manieren tegengaan. Met een SGC-stimulator die vergelijkbaar werkt met stikstofoxide op het ijzergebonden eiwit. En hierdoor de cyclisch GMP-productie kan stimuleren. Tegelijkertijd bestaat er ook een SGC-activator. Die kan werken op het ijzervrije eiwit en daardoor dit inactieve eiwit kan reactiveren om cyclisch GMP te produceren. Ook hier weer twee verschillende mechanismen met hetzelfde resultaat. Meer cyclisch GMP-productie en daardoor hopelijk een beter geheugen. En precies dat is wat ik heb onderzocht in mijn proefschrift. Of SGC-modulatoren het geheugen kunnen verbeteren. Hierbij heb ik gekeken naar het geheugen in gezonde knaagdieren, maar ook naar mechanismen daarachter. Maar hoe meet ik dan het geheugen in gezonde knaagdieren? Dat doe ik met de objectlocatietest. Deze bestaat uit een leertrial en uit een testtrial. En hierbij worden knaagdieren zoals een rat of een muis tijdens de leertrial in een cirkelvormige arena geplaatst waar ze niet uit kunnen. In deze arena zijn op de middenlijn symmetrisch geplaatst twee identieke objecten. De rat of de muis mag deze arena en objecten vrij onderzoeken om zo de omgeving te leren. Wanneer het knaagdier eruit wordt gehaald, wordt na specifiek tijdsinterval het dier ook weer teruggeplaatst voor een testtrial. Alleen in deze testtrial is één van de objecten verplaatst naar een nieuwe locatie. Knaagdieren houden ontzettend van nieuwe dingen. Ze zijn heel nieuwsgierig. En daardoor zullen ze dus ook, wanneer ze de locatie van de objecten tijdens de leertrial hebben onthouden, meer tijd doorbrengen bij het verplaatste object. En dat is precies wat ik meet. Wanneer ik nu 30 minuten voor de leertrial een geheugenverbeteraar toedien, dan kan dit doorgemeten worden in de testtrial. En het tijdsinterval tussen de leer en de testtrial bepaalt vervolgens wat ik meet. Een kort tijdsinterval van 1 uur meet het korte termijn geheugen. Dat is van nature heel goed in knaagdieren en zal daardoor eerst verslechterd moeten worden. Terwijl een lang tijdsinterval van 24 uur het lange termijn geheugen meet. Dit is van nature slecht in knaagdieren en kan daardoor wat makkelijker automatisch verbeterd worden. En precies deze objectlocatietest heb ik gebruikt om een SGC-stimulator te testen, genaamd Riusiquat. Riusiquat is op de markt als adempas voor cardiovasculaire ziekten, voor hart- en vaatziekten, omdat SGC naast belangrijk voor het geheugen ook belangrijk is voor de bloedvaten. Het kan ervoor zorgen dat de bloedvaten zich ontspannen. Daardoor is Riusiquat dus ook op de markt voor hart- en vaatziekten, maar ik kijk dus of deze SGC-stimulator ook iets met het geheugen kan doen. Daardoor heb ik dus eerst een 24-uurs interval objectlocatietest gedaan. Dat wil zeggen, ik heb gekeken naar het lange termijn geheugen, dat zoals jullie zien slecht is in knaagdieren. Wanneer ik dan Riusiquat toedien, wordt het geheugen verbeterd. Het korte termijn geheugen, van nature heel goed in knaagdieren, heb ik verslechterd door neurotransmitter doorgifte in de synapsen blokkeren met biperiden. Wanneer ik dan Riusiquat toedien, dan werken 2 van de 3 dosissen het effect van biperiden tegen. Dit is een heel hersencelspecifiek mechanisme. Maar vervolgens kwam ik erachter dat Riusiquat zich wel verspreidt in het lichaam, maar het bereikt de hersencellen niet. Het is namelijk zo dat de hersenen beschermd zijn van de rest van het lichaam. Niet alles dat door het bloed gaat, komt ook de hersenen echt in. De bloedvaten geven wel voedingsstoffen af aan de hersenen, maar heel veel stoffen worden geblokkeerd door een barrière. En Riusiquat wordt dus ook geblokkeerd. Daarom wil ik dus weten, dit is een hersencelspecifiek mechanisme met biperiden. Hoe kan het dan dat Riusiquat werkt? Dus ik heb nogmaals zo'n korte termijn geheugentest gedaan, maar nu heb ik een andere stof toegediend en dat is een migrænemedicijn genaamd sumatriptan. Dit vernauwt de bloedvaten in de hersenen en daardoor verslechtert het dus het geheugen. Als ik dan Riusiquat toedien, dan werken niet 2 van de 3, maar 3 van de 3 Riusiquat dosissen. Riusiquat werkt dus beter tegen de effecten van sumatriptan dan het werkt tegen de effecten van biperiden. En daardoor kan ik eruit concluderen dat Riusiquat hoogstwaarschijnlijk werkt door het ontspannen van de bloedvaten en zo dus direct dat samentrekken van de bloedvaten van sumatriptan tegen kan gaan. En zo dus het geheugen verbetert. Al met al laat het dus zien dat Riusiquat als SGC-stimulator het geheugen kan verbeteren door het verwijden van de bloedvaten en daardoor een betere bloeddoorstroming in de hersenen. Maar dat de hersencellen zelf niet met een direct mechanisme bereikt en dat daardoor dus ook moeilijk gaat worden om Riusiquat toe te dienen bij mensen die echt hersencelspecifieke ziektemechanismen hebben. Dus ik heb verder gekeken. Toen kwam ik bij een stimulator en een activator, nieuwe stoffen die allebei wel deze hersencellen bereiken. Die komen wel door die barrière heen. Daarbij heb ik ook weer gekeken naar het geheugen in gezonde knaagdieren en heb ik gekeken naar de hersencellen. En vervolgens zie ik met weer zo'n lange termijn geheugentest, weer de knaagdieren van nature niet goed geheugen hebben, dat zowel de stimulator als de activator het geheugen kunnen verbeteren. Maar zoals ik net al aangaf, is er een verschil in de mechanismen. De stimulator werkt op het ijzergebonden eiwit en kan zo de cyclische GMP-productie verhogen. Terwijl de activator werkt op dat ijzervrije inactieve eiwit en dit daardoor kan heractiveren. En deze 2 mechanismen heb ik vervolgens proberen uit elkaar te trekken. Zien of ik dit kan detecteren, dat dit verschillende mechanismen zijn. Daardoor heb ik een stof toegediend die heet L-NAME in de korte termijn geheugentest. L-NAME vermindert de hoeveelheid stikstofoxide dat beschikbaar is voor de hersencellen. En hierdoor verslechtert het geheugen. De stimulator werkt hier wel tegen, maar de activator niet. En dit komt dus omdat stikstofoxide wordt weggehaald, dus dat heeft een direct effect op je ijzergebonden eiwit. Dat produceert geen cyclisch GMP meer. De stimulator werkt hier rechtstreeks op en herstelt zo deze cyclisch GMP-productie. Maar de activator werkt op dat ijzervrije eiwit. En in gezonde dieren is er niet zo heel veel van dat ijzervrije eiwit aanwezig. Dus dan is die cyclisch GMP-productie waarschijnlijk niet genoeg om op te kunnen boksen tegen de effecten van L-NAME. Vervolgens heb ik ook nog naar de hersenen gekeken, ook weer met een objectlocatietest. Alleen dan na 24 uur heb ik geen testtrial gedaan, maar heb ik gekeken naar de belangrijkste geheugenstructuur van de hersenen en dat is de hippocampus. Om te zien of er verschillende mechanismen zijn. Ik heb gekeken naar de neurotransmitterreceptor en ik heb gekeken naar groeifactoren. En dan zie ik vervolgens dat de activator werkt op de hoeveelheid neurotransmitterreceptor die in het oppervlak wordt ingebouwd, terwijl de stimulator juist werkt op het groeifactorsysteem om van zo'n simpele hersencel een complexe hersencel te maken. al met al hebben we dus aangetoond dat SGC-modulatoren de bloedvaten kunnen verwijderen en de bloeddoorstroming kunnen verbeteren in de hersenen. Kunnen werken op het neurotransmitterreceptorsysteem en kunnen werken op het groeifactorsysteem. 3 verschillende mechanismen, 3 verschillende stoffen, maar alle 3 met hetzelfde resultaat. Een verbetering van het geheugen. En met deze conclusie dank ik u voor uw aandacht en geef ik het woord graag terug aan de prorector.
Thank you for your presentation. The opposition will be opened by Professor van den Hove, Professor Epigenetics at this university. Professor van den Hove was chairman of the assessment committee and secretary of this degree committee.
Thank you very much, Mr. Prorector. Let me first of all start by complimenting you and congratulating you on this wonderful thesis, which was a pleasure to read. My compliments also go to the promotion committee. Well, you know me a bit, so we'll also dive into depth a bit, and there might be some nasty questions awaiting you. But anyway, that's part of the job. Let us start maybe with page 75. So it's chapter number 3, where you actually, in the discussion of that chapter, relate your findings in rodents to those in humans. And you actually specifically refer to a study done on healthy volunteers here at our university, the paper by Borgans and colleagues. And then actually you state there, and I quote you, perhaps Rio Chiquat would be more effective in subjects with reduced cerebral vascular health. Looking at your rodent data, I could potentially disagree. Do you have any idea why I could or would disagree on this statement here?
Highly esteemed opponent. First of all, thank you very much for the kind words about both my thesis and also to my promotion committee. With regards to your question, well, I would say there's definitely flaws when it comes to the Rio-Chiquat study. I will not deny that, especially when it comes to the translation of it to the human study. I make some statements with regards to the dose response curve being very narrow, but I would say there's definitely other points to discuss. But I'm very curious, mostly to hear your hint. Yeah,
it's the fact that in the rodents you see actually that even without impairment, you can actually improve memory performance. So you have two scenarios: one where you induce the impairment, and you see whether you can compensate for that. And the other scenario, you actually seem to be stimulating natural forgetting, or the other way around, I should say. You actually prevent natural forgetting. So I was wondering, based on that, whether in the healthy volunteers you might still pick up that effect. Apparently, that's not the case, and that's actually the underlying question. Could you comment on this a bit, or could you think what could explain the fact that you see to have this effect in rodents, preventing natural forgetting, that may not be there in humans, based on the study performed, of course, indeed.
Well, again, thank you for that question. I think the main argument that was also put in chapter 3 here is that mostly we have a very narrow dose response curve, especially when it comes to the natural forgetting, where only one dose of Rio-Chiquat really works in comparison to, for example, the piperidine, in which 2 doses seem to work, if I'm not mistaken. In healthy volunteers, also, there was an impairment with piperidine. Of course, if we disregard the very narrow dose response curve, which of course in animals we give in milligrams per kilogram, in humans we give 1 pill that's based on a 60 kilogram human, but I'm pretty sure there's not a lot of exactly 60 kilogram humans out there, which means that a dose may be missed very quickly, or the effective dose, but despite that, there may be ceiling effects in healthy volunteers. In the end, of course, there's also the problem that it does not cross the blood-brain barrier, and that there is very little known about the peripheral effects on memory. And because there's so little knowledge about that, I wouldn't be surprised if there's a peripheral mechanism that just doesn't translate, because we as humans are more complex and we have a lot of trouble translating rodent studies to humans, especially with regards to the central nervous system. So I wouldn't be able to tell you exactly what that reason would be, but there may be a mechanism out there we do not know about yet.
No, I don't expect you to give the final answer, but I'm happy with the way we're heading. Then let me take a U-turn, so to say, and that actually, for example, concerns page 177. And in this case, focusing on figure 7.1, but actually my question also applies to related chapters and analysis done there. Because I want to talk to you about Western blot. Yeah, maybe you saw that coming.
No.
So I have a few questions there. So a few smaller ones. So why did you hear in panels G, for example, and H, why did you actually express the phosphorylated form of the protein and correlated or corrected that for a housekeeping gene and not for the unphosphorylated form of the same protein? Is there a reason for that?
That's a very good question. I have to dig in my memory. This was done during my bachelor's thesis in 2014. So that's a while back. Definitely one of the reasons is that both phospho-TrkB and TrkB itself have the same species. So they're both rabbit antibodies. In Western blotting, we work with an Odyssey imaging system, which means that we can visualize two different species at the same time as long as we label the secondary antibody differently. In this case, we have the same species, which is a rabbit antibody, meaning that the secondary antibody will always express the same color. So we cannot overlay them in the same blot. So that's why we always have to correct for a housekeeping protein within those blots or between those blots. In this case, the reason why GAPDH was used and not the ratio was because the antibody itself was a little difficult, and unfortunately the outliers within the TrkB and the phospho-TrkB didn't match up, and we would end up with zero data.
I fully understand. So I'm
that's
happy with
not
that answer.
ideal, but
sorry to interrupt. I'm happy with that answer. So a final smaller question then, because that relates then to GAPDH. So any particular reason why you took that one? Because if you look at the literature, that there's papers suggesting that GAPDH is sensitive to iron deficiency, and actually also beta-actin seems to involve in iron metabolism. So did you check whether the housekeeper was actually affected by your experimental manipulation? Yeah, maybe you don't. It's already amazing that you did this work during a bachelor, by the way, so my compliments there. Do you still remember why you chose GAPDH and beta-actin?
Well, the real blunt answer would be because I was a bachelor student and my supervisor told me to. But let's.
I think that's a valid answer.
But let's hypothesize for just a second. I will try to not make this too long. I think I researched it in hindsight, and I don't think there's a very ideal housekeeping protein anyway. If we look at Western blotting, the best case in total protein samples is to go also for total protein analysis, which is the future, and unfortunately we have not really implemented in our lab yet, and I know that our technician is working very hard on that, and I'm very happy with that. So that's a flaw, and you're correct, and that should change in the future.
Okay, I'm very happy with your answers, and I give the word back to the program.
The opposition will be continued by Professor Kessels, Professor Cellular and Computational Neuroscience at the Amsterdam University. We highly appreciate your participation at this ceremony, and Professor Kessels was also a member of the assessment committee.
Thank you, Chairman, and dear candidate, I also want to first congratulate on your really nice thesis. I also enjoyed reading it a lot. You know, it's also one of my favorite topics, of course, but it's really well written and a lot of work. So also my compliments to promoter and co-promoters. But without further ado, I want to ask you, actually, I want to start with asking you a bit about the BDNF and pro-BDNF. Right, so this system is used in our brain, and it has effects on synaptic plasticity. So how would you predict the levels of BDNF and pro-BDNF would affect memory formation?
Highly esteemed opponent, first of all, again, thank you very much for the kind words about my work. With regards to the pro-BDNF and the BDNF in memory, it's a complex signaling pathway. Generally speaking, we see a sort of yin-yang effect. I think that's also one of my propositions, where a mature BDNF is usually inducing neuroplasticity and therefore important for memory formation, whereas pro-BDNF, when binding to the P75 neurotrophic receptor, is considered apoptotic and therefore inducing, well, the opposite. So that would be bad for memory, to state it like this. It's a bit more complex than that.
So I can actually go on, because I agree with you. BDNF would promote plasticity, and pro-BDNF would inhibit it, maybe even cause synaptic depression. But then I also go, I'm also going to ask you to dig deep into your memory. I'm going to go to chapter 7.
Oh dear, sure.
Because in your discussion, you then say that actually, because this is a study in piglets, right? They have reduced amounts of iron in their diet when they're very young, and then later you're going to look at their brain and hippocampus, for instance, and see BDNF and pro-BDNF levels, and then see that there's actually a negative correlation, you say in the discussion, between BDNF levels and memory formation. So if BDNF is promoting plasticity, how is that possible?
That's a very interesting and complex question, I'd say, and it also really broke my brain a little bit around that time. I will admit. We also found very different results for the ventral hippocampus versus the dorsal hippocampus, which was very unexpected.
So did you also do a correlation between the learning behavior and the?
I think we did, yeah. I think we did, but it's long ago.
Right.
But definitely, to come back to your question, when it comes to BDNF being negatively correlated with memory performance, I think the argument I gave in the paper itself was that we're in a neurodevelopmental stage where it's not just long-term potentiation and synaptic plasticity we want to promote, but there's also a very strict orchestration of neuronal apoptosis, so we don't have an overexcited system, so to say. That's the argument I give then. I don't think I can give you a better explanation than that, because I simply didn't know. I still cannot really give a clear answer on it. I know that pro-BDNF is very complex. It's very under-researched, if I may say so. And if it binds to TrkB, especially when TrkB is phosphorylated and dimerized, it can actually also induce neuroplasticity. So it's not all black and white, and I think that's the most important thing in the discussion.
Right.
If that answers your question.
You mean it's difficult to make a conclusion? For instance, it could actually be that these different BDNF, pro-BDNF levels are a reflection of a poorly developed brain.
Yes.
Right. Maybe there's miswiring going on, and that's
exactly. That was also a bit what I was trying to say, but definitely not clear.
I also want to have another point of this same thing. This is kind of, I'm still talking about chapter 7,
yes.
Back in your bachelor days, figure 1. But this is kind of a general question.
Course,
but it's also reflected in this figure, because in figure K you look at PSD95 levels, and then, for instance, in the ventral hippocampus you see 3-fold increase in PSD95 levels, right? Mm-hmm. So the bars are 3-fold higher, but it didn't get a star. As in statistical, there is no significance. True. For instance, and you look at H, there's almost the same graph, and it did have a star. And then the conclusion, what we always do in science, is if there's a star, we say it's different, and if there's no star, we say there's no effect. How do you feel about that? Should we ignore the bars and just look at the stars, or?
We should definitely always try and. Oh, this is going to sound so terrible. Search for the stars. But no, of course, we need to rely on statistical significance, but we're always also at some point dealing with type 1 and type 2 errors. I cannot deny that this is slightly strange. I cannot deny that the phosphorylated TrkB antibody is not the best in the world. And perhaps if I have to choose which data I stand for the most, it would definitely be the PSD95 one. I think is a power thing. It's a power issue. We had some I would say, how do you say it? Outliers, technical outliers mostly, because we had a broken Odyssey imaging system around that time. The phosphate TrkB was actually not done by me.
So this is all specific to this particular experiment, yes. But in general, do you feel we take a statistic too serious sometimes?
Not necessarily. I think we shouldn't fare fully on statistics. We shouldn't fare fully on gut feeling. Sometimes statistics are very strict, especially when it comes to large genome data. Sometimes the corrections with regards to p-values becomes so strict that you're going to miss effects even if they are there. But if we stop using statistics at all, we can just work on absolute effects and go, well, that looks nice. So there should be a golden ground somewhere in the middle. And yeah, that's my take on it. And we should mostly rely on statistics anyway. We have to have some kind of guideline in our work. That's my final conclusion on that, I think.
Yeah, you're kind of nicely expressing my frustration generally with statistics also. For instance, if I would repeat this experiment and get the same graph, but it is significant, my result would match your data, but would contradict your conclusion, right?
That is true. Well, yeah, that would be true. But then a third experiment, one is always an incident, two could be a coincidence, and three is a pattern. So then if two things do not show the same, we should always do it a third time, and then we'll know for sure.
Okay, so
but it could contradict. Well, contradict the conclusion is not maybe the best word, because we don't say there's an increase in PSD95, but we don't state that there's no plasticity.
But you do conclude there's no effect in PSD95.
Exactly, but the overall effect is maybe not as strong. Or the overall conclusion is not as strong, but yeah, the conclusion might change. That's true, and that's a flaw. That's definitely the case.
Which we all have to live with, actually.
Yes, and that is especially a flaw in Western blotting, specifically, I think, because it's so semi-quantitative.
Right. So maybe with our conclusions, we have to sometimes be a bit more nuanced. I think that's what you wanted to say.
Yeah, I think that's a good summary. Yeah.
Okay. That's a very satisfying answer. Thank you very much.
Thank you. The opposition will be continued by Professor Knipper, Professor Molecular Hearing Physiology at Tübingen University in Germany. Professor Knipper was member of the assessment committee, and we also highly appreciate your participation to this academic ceremony.
Yeah, so I hope you can understand me. So I also wanted to start, Ellis, with a congratulation to this very important and very excellent work. The relevance for the society cannot be underestimated, for my opinion, because of the high impact cognitive diseases may have in the future. And congratulations also to the supervisors who guided you through this very complex topics. So perhaps I would, although I would love to continue with the discussion of Professor Kessler, I will start with my first main question, which addresses your interesting finding that you found that riociguat reversed short-term memory and long-term memory effects. A feature, so it reversed these short-term and long-term memory effects when it's impaired differentially by your drug, as you did show, and it did it differentially, and you discussed this in several of your chapters as a feature that is interpreted by vascular peripheral effects of CGMP signaling, but not direct on the brain. Can you explain what you mean with this?
Highly esteemed opponent, first of all, thank you very much for those lovely remarks about my my thesis there. It's very humbling, actually. Thank you very much. With regards to your question about cyclic GMP signaling and what I mean with the peripheral effects, so indirect effects, yeah, if I understood correctly.
Yes, you understood correctly.
Okay, perfect. So riociguat, but also vericiguat, do not cross the blood-brain barrier, so they don't have effects on neurons immediately. So everything they do should be exerted via the blood vessels or any other indirect mechanism. That's what I mean. That doesn't mean that it could, for example, to give an example, they could have an effect on the endothelial cells or the vascular smooth muscle cells in the cerebral vasculature, which through neurovascular coupling could have an indirect effect on the neurons. Not just the neurons, also the glial cells. I should not forget glial cells. They're very important, especially in the blood-brain barrier. And that's the broadest answer I can give. That's the broadest answer I can give, but you're asking me to specify.
So if you can specify what you really mean, in particular to some other surprising effects that you saw with this drug, no direct effect, I presume, if I memorize correct, on the LTP, on AMPA receptor trafficking. What is your idea behind? I will help you later on.
Okay, my idea at the moment is that there's a small vasodilation of the microvasculature. So when it comes to nitric oxide, this could be capillaries through, for example, pericyte action or more arterioles. But I think there's, especially on the microcircuitry level at certain spots in the brain, I think that there is a slight vasodilation that's just enough to boost the blood flow ever so slightly and improve memory. This is also why I think we don't pick it up in the MRI we did with Vericiguat, the data there with the data in chapter 4. I don't know if I cannot recall the page right now, but I think especially because we don't see that effect on whole brain cerebral blood volume. It's specifically so a vasodilation of the micro circuitries, for example, really located to a hippocampus or something similar.
Perhaps I can go once more in, because we nasty questions are also allowed, I heard.
Of course.
You mentioned this indeed, what you just said. So one of the surprises, probably for you as well, was that it was very Siguat, which you show, although it affected ex vivo memory and whatever, but it did not change blood flow. How do you explain this? So because so the cerebral blood flow volume is not changed, correct?
That is correct.
So although the whole idea of NOGC stimulators is somehow based on neuronal, or is it not also that the people still think that NO is released from endothelial cells and you would expect that it changes the blood flow?
In the case of Vericiguat, I would definitely say the latter, especially because, to go back to Vericiguat, we find an effect on PVASP. And this is most likely endothelial or vascular effects. And that shows that at least in the cerebrovascular cells, there is something happening with NO signaling and with PKG activation. So I would say that the latter, definitely, from what you just mentioned. I think I lost part of your question. I don't think I answered everything.
My question was,
yeah.
Do you have, you answered already part of this. The question was, what is your idea about neurovascular coupling, which you may influence? This was the aim, was it? So you mentioned in your thesis that, to the surprise, AMPA receptors were not. So my question is, I make it more precise. Have you heard a role of inhibitory input on vascularization? And can you exclude that your drug changes inhibition?
I can for sure not exclude that the drug changes inhibition, especially because, well, in this case, it would be especially important for brain penetrant compounds, but it's known that GABAergic provalbumin, GABAergic interneurons actually produce nitric oxide through nNOS. So, and this is important for the inhibitory networks. So I would for sure cannot exclude that the drug would not do that. Of course, in this case, GABAergic interneurons in the central nervous system would not be directly influenced, but if eNOS produces nitric oxide, it can freely distribute. So it can reach still some neurons in the vicinity, probably not to an extent that we can measure it with a Western blot. But it might still be possible. And to come back also to figure 4.4, I found another page in the meantime. There is another thing with the figure, which is that the doses are the dose administration route, which of course is necessary for this method, are not identical to what we did with memory. So there may also be a discrepancy that would explain why we maybe do not see anything with periciguat. I wanted to highlight that still. So it may actually be possible that there is an effect on cerebral blood volume that we miss here because dose translation wasn't 100% perfect. So that is important to highlight, I think.
Mm-hmm. Perhaps a last brief question to the BDNF. You discussed the BDNF in the context of a neurotrophic factor and memory. Have you heard that BDNF is expressed in capillaries and in platelets?
Yes, that's, yeah.
Have you heard, and did you consider perhaps if what kind of, in case, I do not know if you have heard it, BDNF is discussed in the context to play a role above the role which you currently discuss in the paper, that it also may influence the vascularization. And your drug effect of BI-747 in comparison to Runca-Ciogat, which both enhances, if I'm right, AMPA receptor mobilization and LTP, but the effect of BDNF is a little bit different, if I can recall. So can you consider, or did you consider a role of BDNF which is independent of a direct effect of neurons?
That's a very interesting suggestion. I can say that I did not consider this, otherwise I would have also put it in the discussion. So that would have been a very important point. And to hypothesize about this a little bit, if this effect would indeed be there, it would be an even stronger argument why a compound such as BI-747, or to a lesser extent Runca-Ciogat, which is a bit more on the neurotransmitter system than on growth factor, but still also acting on mature BDNF. Why compounds such as these would be so great for cerebrovascular disease, such as vascular dementia, vascular cognitive impairment, because we could, and that's my philosophy from the thesis, actually, we could then be tackling two systems that are somehow both intertwined and both diseased and influencing each other in a negative way. And if we can tackle both from a vascular perspective with these compounds, and it penetrates the brain, and we can affect the central nervous system perspective, that sounds like a bit of two of a perfect world, actually, but that would be a perfect world. So I would hope, actually, there's indirect effects, and this is something that I should be measuring in the future.
So I would motivate you also. So all my questions are addressed. Thank you so much. Ellis.
Thank you very much. The opposition will be continued by Dr. Casas, Assistant Professor at the Department of Pharmacology and Personalized Medicine at this university. She was also a member of the assessment committee.
So first of all, I want to, of course, congratulate you for really a huge amount of work. The thesis is quite complex and quite long, and it's full of really nice data. So congratulate you for your thesis first, and of course your promoter and co-promoter to go with you in such a long and tough process that we really know about. So after doing that, I want to somehow continue with my first question a little bit with the topic that Professor Nipa was just mentioning about the vasculature. So I somehow came up with the same situation in terms of that you claim a lot about a vascular effect, right, along your thesis in different chapters, in discussion, in introduction. But somehow when you go a little bit to the vasculature, the only data that you have, it's not really that apparent, because the MRI data that you have, it's not really too surprising. So I wanted to know in which direction you want to go to really prove your hypothesis, because I have the feeling that it's a nice hypothesis, but in your thesis was not completely described. So are you already doing, have you done already experiments? Are you planning to? What is the status there? Because I'm really curious about in which direction actually you're handling this problem.
Esteemed opponent, first of all, again, thank you very much for the kind words and the acknowledgement of my work. With regards to your question, I would first like to agree that indeed I make some strong statements, especially about the vasculature, that are maybe not completely supported yet. And I did this purposefully so, because I think we should look at this more. And that also means that I am planning to do more in the future. My current work actually is looking at individual cell types, looking at both in vitro, so looking at neurons, microglia, astrocytes, and endothelial cells, and seeing if I can model aspects of vascular cognitive impairment in there, but that would still not be 100% vasculature, maybe the endothelial cells to some point. But I'm also planning in the future to work more in vivo, both chronic treatment in healthy mice, and then using blood flow measurements such as laser speckle, or for example, or Doppler imaging to look at local blood flow and the effect of SGC modulators on this chronically, and then hopefully further along the line also using a model for vascular impairment, such as, for example, the bilateral carotid artery stenosis model that Dr. Fouquier has actually developed in Maastricht. So I really would like to test my modulators in there. Of course, this is a long process. It's going to be ethically also challenging to get everything approved, but this is for now also part of a grant I was awarded. So I am planning to do this still, and I hope that this will give me a lot more insight.
Okay, yeah, nice approach and nice answer. Thanks a lot. So based on my background and so on, my next question is going to be, of course, more focused on translation. And the majority of your experiments, you give actually the treatment around 30 minutes more or less before the test, right? With 24 hours in the almost all experiments interval. So how do you think that you can actually, such a super short period, how can you translate such a therapeutic scheme to the clinics? So how could you think that that would be feasible for a patient?
Well, thank you for that question. I would say that particular acute test is maybe not immediately translatable to a patient. This is more a concept. Can we enhance memory when you administrate a compound 30 minutes before learning? We look at acquisition processes, which are quite fast. They're fast processes such as neurotransmitter release, such as trafficking of receptors. And then we prove that, yes, the compounds have an effect, but on the long run, we would have to do, depending on what the eventual goal is going to be, preventative or treatment, as in after an incident of some sort. So this will also have to be done with chronic treatments as a preventative measure, or in vitro. I'm actually specifically looking if after I induce an inflammation or oxidative stress in my specific cell types, can I then still rescue all of the phenotypes I see? The same will be done in vivo when we use, for example, that stenosis model. This will not just be preventative, but especially after the surgery has been done, can we rescue? Because that's more of a logical clinical translation than just giving a drug once and then assuming that it's going to help a patient.
Yeah.
I hope that answers your question.
That's exactly the answer that I was looking for. It was, of course, not a question to say I would translate it directly. I was not expecting that. I was expecting how you would do it. Okay. And that's exactly how your question, or in the direction of your answer. was. Okay, so sorry if it was a little bit of a trap. I hope not. It was more to see your opinion about it, like how you would do it. So you already actually answered my next question, that it was about disease models. You already mentioned this a nice model about the occlusion of the carotid artery and so on that you already mentioned. So you already answered that. So my final question would be about the compounds. So you mentioned, or you touch upon several compounds in your thesis. You touch upon riociguat, bericiguat, Bay 747, and so on. So which one do you really think that has the highest potential in terms of clinical translation, and why?
A short answer, please.
At this moment, if approved for humans, roncaciguat.
Okay.
Can I explain? No? Okay.
Okay, thank you. The opposition will be continued by Dr. Foulquier, assistant professor at the Department of Pharmacology and Toxicology at this university. He was also a member of the assessment committee.
Thank you, Mr. Porter. Well, first of all, dear candidate, I would like also to compliment your work and nice thesis and that you have achieved with your promotion team, and also the nice presentation you gave us today. Actually, I'm going to start also with a question about patients, because indeed your work can lead to very promising outcome for patients with vascular dementia. And that's something you discuss in your thesis, obviously, especially focusing on vascular cognitive impairment and dementia. I was wondering, because the patients coming at the cognitive clinic, when they have cognitive complaints, their cerebral blood flow is not being measured at the moment. So now they get an MRI and they get okay, I understand that they have some lesions in the brain, many different lesions that you also describe in your thesis, white matter lesions, enlarged perivascular space, lacunar infarct, and so on. So they are all different, they can present different lesions. Do you think that a single therapeutic approach would still be eligible for them, or what is your take on that? Would you actually give them rather a stimulator, an activator, how to actually enhance their cognitive function?
Well, esteemed opponent, first, thank you again very much for the kind words, but also for the tough question. I think that's the big question here. Would an activator or a stimulator be better? First of all, when it comes to the diagnosis, I think we must remain with a multi diagnostic approach, or use multiple means to determine, or hopefully determine, some kind of mechanism that's behind it, because vascular cognitive impairment is a very large term that involves everything that induces cognitive impairment resulting from a vascular perspective. That can be vascular dementia, that can be Alzheimer's with a vascular component, to give two options. So the mechanism of disease is going to be very important. So we need to keep that in mind, and that may also have an effect on whether a stimulator or an activator is going to work, because it may be different, especially when it comes to stroke. Dr. Casas, for example, has shown beautifully in her work that there is, after stroke, or after ischemic stroke, we can detect a lot of apo-SGC, which would suggest that immediately an activator would be a good idea. But on the long run, there could be question there. So to wrap it up here, maybe a combination of both in the correct dosing, depending on the underlying problem, may be actually the way to go.
Thank you for that answer to that very difficult question. Indeed, I think no one has the answer, so indeed the more research is needed there. Actually, yeah, I'm not going to ask you a question about angiogenesis, because it was also slightly touched upon by our previous questions, but that may be something we might have to discuss in the future. But maybe I would like to rather instead come back on something you mentioned in one of your earlier answer, because you actually described this pathway, the leukocyte guanylate cyclase pathway for the neuronal compartment, for the vascular compartment. But as you also said today, glial cells are of importance here, and this neurovascular coupling is quite recent. So I would like to know, yeah, what is already known on this topic? And do you think that targeting also these cells would be of relevance somehow for vascular cognitive impairment?
That's a very good question. I would say yes, it's very important to look at astroglial cells, to call it as a whole. I would also say there's not a lot of literature on it at all when it comes to specifically SGC. A lot of people look at cyclic GMP, but we must also consider that cyclic GMP and PKG both can be also derived from particulate guanylate cyclases. So we can never assume that whatever we find on cyclic GMP can be translated to SGC. There's not a lot out there. When it comes to astrocytes, a little bit is known about the fact that it does have an important SGC signaling pathway, and actually in Alzheimer's brains, it was specifically found that SGC protein expression was SGC expression, should not say protein expression, SGC expression was reduced in specifically astrocytes. But because of the difficulties investigating all of this, because we didn't really have any brain penetrant compounds until, I would say, the first publication in June, actually, by Cyclarium, unfortunately, if I may say that. We didn't have them. It's difficult to investigate this in an actual functioning living system. So there's nearly no literature, and that's unfortunate, and we should change that.
Thanks for your answer. I think due to sake of time, I think I will give the word back to the proctor. Thank you.
Thank you. The opposition will be continued by Professor Bronkaerts. Professor Bronkaerts is working at the Laboratory for Research in Ischemic Stroke Stem Cells and Angiogenesis at the Hasselt University in Belgium. Also for you, thank you for coming and joining this ceremony.
Dear Ellis, it was a delight to read your thesis, and also the artwork I could really appreciate. It was something different. So now I also have a question. So you really see that some of your compounds do not need to enhance, to enter the brain, and they probably work via the blood vasculature. So what if you would use your compounds in older animals who have a more stiffer vasculature and who have less NO coupling? Would they still work?
Esteemed opponent, first of all, thank you very much for the lovely compliments. This question, it's very coincidental. I was actually, when I was preparing for my defense, the same thought popped into my head. I'm not making a joke here. It's actually true. I thought, we never looked at aged animals.
Because that's where you want to win. You want to do something about dementia. So that's the area where you should
that's very much true. And I actually think that might be a good extra experiment in some way, maybe somewhere in the next 2 years to wiggle in if I can find the time. But it's definitely important to start looking at this, especially if we consider that a blood-brain barrier integrity is not going to be perfect in older animals. And maybe we can actually have a little bit of leakage of compounds such as riociguat or vericiguat inside the central nervous system, which is then going to even enhance their effects. So just hypothesizing here, of course, but I agree with you. And older animals might be a very good idea, indeed.
Yeah, because there's maybe more to win. And also you say academia and pharma should consider that brain penetrance of these GC stimulators may actually not be required for an effective treatment. But I was thinking, if you have stroke patients or patients with dementia, do they then need other doses of the treatment? Because they have leak BBB, maybe then the compounds can penetrate the brain. What about that?
That's something I also thought about, actually. And it's a difficult one. I put that statement, that proposition in because I wanted to be bold, and I realized that it's very difficult to publish things when a compound's not brain penetrant and it works, and it frustrated me because it works, and especially in older animals and in diseased animals, the blood-brain barrier the integrity is reduced, and we should not always go, okay, it doesn't cross, so it doesn't work. We have enough translational issues as it is. So I got a bit lost there on your question. I got a bit enthusiastic about my point here. Help me.
So you want to investigate that, let's say.
Yeah, definitely.
And then I want to come to another point. So I was reading your thesis and I was thinking, wow, how interesting. Why doesn't she combine the GC activator with the GC stimulator? Would that make sense? or not?
Yes, I think that would make sense. And now I remember the dosing question. Okay, because that brings me back to the dosing as well. There may be a difference in dosing needed then. I also quickly said it to Dr. Fulke already, and it also may be, by the way, with regards to, for example, vericiguat, we don't really know which dose would be good for the vasculature, which dose would be good for the brain, and especially with activators and stimulators, given that they have, if we look at BI-747 and runcaciguat, different effects on different parts of the cell, maybe because of compartmentalization, maybe because of different binding kinetics or whatever. It doesn't. They have different effects, and depending on the disease mechanism, it may be nice to boost something there and something there. And there, for that, we would need dose response curves, combination treatments. It would be a lot of work, but it would also be very important work to look at. So that's definitely something that I am planning to look at in the future, because why use only one of them when the combination may be better? Because there is NO scavenging, but there's also oxidative stress. So we have less active SGC because there's not enough NO, but we also have less active SGC because there's no ferrous heme group anymore. Why only push one system? It doesn't make sense. At least to me.
I wouldn't say, when I was reading, it makes sense. I was thinking, oh, why don't combine them?
No, to me, it doesn't immediately make sense, but not everything has to make sense to work, so that I should always keep in mind. But yeah, I would love to combine it at some point and see if that would actually work better.
Okay. Can I have one more question? Or? Yes. And then to wrap up, I want to end with number 10.
Oh dear.
Le meilleur travail n'est pas celui qui te coûtera le plus, mais celui qui te réussit le mieux. So the best work is not what is most difficult for you, it's what you do the best. I totally disagree with this. How will you grow? You never grow inside your comfort zone.
That's true, but you don't have to stay within your comfort zone to realize that some things you're just not talented for. I mean, I am a big Sartre lover. There's people that are very much against it. So that's definitely a discussion to be made. For me, this doesn't mean you shouldn't step out of your comfort zone. It just doesn't, it means that you shouldn't be, as we would say in Dutch, kick to a closed door too much. That's a terrible translation, but you know what I mean. We should always look for our own strength and not try to challenge our weaknesses and then get lost in this consistently. And with that, I guess I would have to end.
Ellis Nelissen, de gelegenheid voor het verdedigen van uw proefschrift is voorbij. De promotiecommissie zal zich nu voor beraad terugtrekken en ik verzoek u en uw gezelschap onze terugkomst en de uitslag van dit beraad in deze zaal af te wachten. Ellis Nelissen. De commissie hier aanwezig heeft zich beraden over de kwaliteit van uw proefschrift en de wijze waarop u dat proefschrift heeft verdedigd. Op grond van haar positief oordeel en gelet op de examens die u vroeger heeft afgelegd, heeft zij besloten u het gevraagde doctoraat toe te kennen. De hoogleraar, professor Prinkaerts, is gemachtigd u op de gebruikelijke wijze met deze waardigheid te bekleden. Het woord is aan uw promotor.
Dank u wel, meneer de prorector. Allereerst de eed. Belooft u dat u altijd volgens de beginselen van wetenschappelijke integriteit te werk zult gaan, eerlijk en zorgvuldig, transparant, onafhankelijk en verantwoordelijk?
Ja, dat beloof ik.
Dank u wel. Krachtens de bevoegdheid ons door de wet toegekend, volgens het besluit van de commissie hier tegenwoordig, verklaar ik hier bij u, Ellis Nelissen, tot dokter te bevorderen en u alle rechten te verlenen die daaraan volgens wet en gewoonte zijn verbonden. Ten bewijze hiervan ontvangt u binnenkort de bul door rector, secretaris en promotor ondertekend en met het grootzegel van de universiteit bevestigd, zoals nu getoond door mezelf. Proficiat.
Dank u wel.
Dan zijn we nog niet klaar. Dan heb ik ook het genoegen om jou nog een certificaat uit te mogen overhandigen. Dat zal Dr. Peter Sandner van Bayer doen, het menscertificaat. Peter?
Yes, thank you very much and congratulations from my side. Very nice work and happy to give you this PhD certificate.
Thank you very much. This was the certificate from the Institute. Now the Institute from the Uran Graduate School, delivered by Dr. Tim van Mierlo.
So congratulations also for my side, Dr. Nelissen. I have the honor to give you or to grant you the Uran PhD Certificate of Excellence for fulfilling all the criteria which adhere to this certificate. So congratulations.
Dankjewel. Ik ga toch naar het Nederlands weer? Oké, dan is het nu tijd voor de laudatio. Beste Alice, allereerst gefeliciteerd. En ik wil bij de felicitaties ook jouw ouders betrekken. Proficiat. Ora est, dat is zover. Je bent dokter nu. En aan mij de eer voor de laudatio. Dan gaan we meteen naar het begin. 2014, lang geleden. Je liep toen een bachelorstage in mijn groep als student biomedische wetenschappen. Achteraf, ook om strategische redenen, om jouw kansen te vergroten om aangenomen te worden voor de specialisatie Fundamental Neuroscience, ook bekend als FN, van de Research Master Cognitive and Clinical Neuroscience. Dit is in mijn optiek misschien wel de beste master van deze universiteit. Sommige mensen weten dat. En het is niet gemakkelijk om hier binnen te komen, maar bij jou waren we het snel eens. Geschikt. Dus tijdens deze master leerde ik je nog beter kennen, ook als studentvertegenwoordiger van jouw jaar, van jouw cohort. En een hoogtepunt voor mij was de oprichting van de FN-band, met alleen maar studenten van deze specialisatie. Deze band speelde in 2016 op het symposium ter ere van het 10-jarig bestaan van de master. Jij speelde in deze band uitstekend gitaar, dus dat kun je ook nog altijd gaan doen na de wetenschap, mocht het nodig zijn. Ik was trouwens de drummer, je kunt mij ook huren. Goed. Later kwam ik erachter dat je ook basgitaar en piano speelt, dus je hebt heel veel hobby's blijkt. En fotografie is er ook nog eentje van. En dat zie je ook denk ik in die mooie cover, dat je echt artistieke kwaliteiten hebt. Wat heel goed is voor een wetenschapper, denk ik. In 2017, ja, dan gaan we naar 2017, heb je jouw stage gedaan in een lab van de beroemde professor Bading in Heidelberg, Duitsland. Daar heb je heel veel geleerd over celkweek, maar het meeste heb je geleerd over jezelf. denk ik. Het was niet gemakkelijk in het begin. En met doorzettingsvermogen heb je geleerd dat je jezelf kunt redden in een nieuw lab en in het buitenland. Wat mij betreft misschien wel de belangrijkste stap van jouw carrière. Eind 2017 zocht ik iemand om experimenten te doen voor de firma Bayer. Hier aanwezig in de persoon van Dr. Peter Zandner. Ik werkte al een tijdje voor Bayer, of niet voor, met Bayer. En we hadden al mooie in vitro en in vivo experimenten gedaan, waarbij we SGC-modulatoren hadden getest. Dat was nog werk van Nick van Goetem en Inantine Argeruzzi. En Bayer wilde nog enkele in vitro studies doen en het was mij meteen duidelijk dat jij de aangewezen persoon hiervoor was. Lang verhaal kort, je hebt die experimenten uitstekend gedaan en uitgevoerd in Hasselt, trouwens. En hierover een heel mooi rapport geschreven en ik wil ook Tim ook nog eens bedanken voor de support en steun tijdens het hele promotietraject. Ondertussen was ik helemaal overtuigd van jouw kwaliteiten en had je ook mij overtuigd dat je PhD-student moest worden. Je had het zelfs gepresteerd om ook al in 2017 jouw stagewerk als student te publiceren. Dit is hoofdstuk 7 in het proefschrift, waarbij breinplasticiteit in het varkensbrein werd onderzocht. En tot haar schrik werden er heel veel vragen over gesteld. We besloten om een proefschrift te maken over de relatie tussen stress en plasticiteit. En in april 2018 werd je PhD-student. We hadden in 2018 al snel 2 artikelen, 1 over stress en geheugen, 1 over postpartum depressie. En in 2019 kwam hier een MSIS-paper bij, ook bekend als het COTS-paper. Hierin beschrijven wij een diermodel om de bijwerking overgeven van medicijnen te bestuderen en te testen. En een van die medicijnen was een PDE4-remmer. Ondertussen had zich een andere Duitse firma gemeld, niet Bayer, met een plantenextract dat PDE4-remming liet zien. En of wij dat wilden testen. Nou, dat wilden we wel. En we besloten alles om te gooien. Het proefschrift zou PDE4-remming en plasticiteit worden. We hadden nog tijd genoeg en gingen ervan uit dat we wel zouden mogen publiceren in ongeveer 2 jaar. Helaas bleek het extract niet geweldig te werken. Het waren pittige en langlopende experimenten. En toen besloot de firma ook nog eens dat we niet mochten publiceren, voorlopig. In ieder geval niet voor het proefschrift, behalve de data van hoofdstuk 6, waar niemand wat over vroeg. Maar wat nu? Ik herinnerde me ons Bayer-werk en heb Bayer toen, Peter, gevraagd of we misschien niet al mogen publiceren. En tot mijn grote vreugde, onze grote vreugde, mocht dat. Sterker nog, we werden gestimuleerd en ondersteund om nog aanvullende experimenten te doen en de artikelen nog beter te maken. En vrij snel, heel snel, hadden we 3 mooie manuscripten die geresulteerd hebben in hoofdstuk 3, 4 en 5. Daar zijn we Bayer heel dankbaar voor. Thank you, Peter. I hope you understood it, but thank you. In oktober 21 was je klaar met je proefschrift, dus je hebt alles in 3,5 jaar gedaan. Topprestatie. Ook tijdens deze jaren heb je veel geleerd, dat zal ik even opsommen. Allereerst het doen van uitstekende wetenschap, dat hebben we net kunnen zien. Breed scala van technieken, in vitro, in vivo, moleculaire biologie, gedrag. Dus dat kun je heel goed. Daarnaast heb je geleerd om, heb ik opgeschreven, het vliegtuig te pakken. In 2019 hadden we een Euromens, waar je ook certificaten van hebt gekregen, workshop, psychofarmacologie in Heraklion, Kreta. Dat was dus duidelijk voor COVID. En toen kwam ik erachter dat jij nog nooit gevlogen had. En als zo vaak bij jou, je vindt iets spannend en achteraf is het de normaalste zaak van de wereld. Je paste je ook moeiteloos aan het strak congresschema van overdag wetenschap en 's nachts uitgaan. Dan stip ik ook nog even aan het schrijven van werkprotocollen en alles wat erbij komt kijken. Dit zijn protocollen om goedkeuring te krijgen voor proefdierstudies. Dit is echt heel veel werk. Nederland is op dit moment het strengste land voor proefdieronderzoek. Maar dankzij Ellis kon het meeste proefdieronderzoek in mijn groep doorgang vinden. Waarvoor dank. Dan het begeleiden van reguliere studenten en Braziliaanse PhD-studenten. Op de een of andere manier trek je mensen aan die gemiddeld meer aandacht nodig hebben. Met engelengeduld heb je al deze studenten geholpen en beter gemaakt. Waarvoor dank. Wat ik ook zeer op prijs stel, is dat je mijn favoriete hersenstructuur op de voorpagina hebt gezet. Dames en heren, dit is geen zebra, dit is een zeepaardje, ook bekend als hippocampus. Een zeer belangrijke hersenstructuur voor het geheugen. Dan ga ik nu afronden. Beste Ellis, ik kan je omschrijven als direct, hardwerkend en loyaal. Je bent zeer kritisch naar jezelf. Aan de andere kant ben je ook wel eens zeer kritisch naar anderen, wat geen slechte eigenschap is in mijn ogen. Je bent altijd bereid, soms niet meteen, maar uiteindelijk wel, om collega's en studenten te helpen en hun problemen op te lossen. Je werkt snel en met grote precisie. Je bent een fast learner, ik vond geen Nederlands woord, fast learner, en mijn PhD-student met de beste work hard, work smart mentaliteit. In feite heb ik vergelijkbare woorden gebruikt als aanbeveling voor de coachschapssubsidie die je hebt gekregen. Dit is een fantastisch vervolgproject op jouw proefschrift. Eveneens weer in samenwerking met Bayer. En ik ga dan nu ook afsluiten met mijn aanbeveling in het Engels van de Coachra-aanvraag. If this postdoc project yields positive data, it could be the start of a shiny career with her as one of the pioneers in the field. I would like to point out that the entire proposed postdoc project comes from her, and she therefore deserves to develop her innovative ideas further. Also because, in my opinion, she has the qualities to make it happen. I expect a lot from her. Alice, dan sluit ik nu traditioneel af met: je kunt heel trots op jezelf zijn. Proficiat.
Dr. Nelissen, namens de universiteit wil ik u ook van harte feliciteren met uw nieuwe waardigheid als dokter. Tijdens de verdediging is al duidelijk het belang van uw onderzoek eigenlijk onderlijnd en de verwachtingen die in u gesteld worden zijn net door uw promotor uitvoerig beschreven. Wij verwachten inderdaad als universitaire gemeenschap zeer veel van uw onderzoek en het Coachra Fellowship is denk ik daar een ideale voorbereiding toe. Als laatste, u hebt al een aantal certificaten ontvangen. Ik ga u nog het certificaat geven wat door de rector zelf eigenlijk ondertekend is. En als blijvende herinnering, en ik denk dat dat wel past bij jou, overhandigt de universiteit die ook graag het klassieke beeld van Maastricht, de Maastrichter Gijs, zodanig dat je een blijvende herinnering overhoudt aan deze dag. Ik wil nu vragen dat de genodigden het auditorium verlaten. Ondertussen zullen wij vooraan een foto maken samen met de online collega. En hiermee sluit ik deze academische zitting.